Okay, so you know what genes look like in prokaryotes, but what do genes look like in eukaryotes? Well, they're more complex, they're bigger, and I'll tell you all about it. So, what's not different? Well, it's not different in the way that we decorate them, and we decorate them using, again, the coding strand. And because we have a double-stranded DNA genome, that means, again, we have a template strand. So the RNA polymerase will read the template strand and write in a pre-mRNA this time, pre-mRNA that looks like the coding strand. Okay? So what does that mean? That means right here we have a promoter. This promoter is complex. It's made up of multiple parts. Talk about those parts in a minute. Okay? Outside the promoter, what do we have? Well, we no longer have parts of the gene, parts of this transcript encoding multiple genes. It now only encodes a single gene. So these, rather than genes, these boxes are exons. Exons are parts of the mRNA that will code for proteins. And we'll make a single protein from all these exons together. In between the exons, we have introns. So this would be intron 1. This would be intron 2. That makes this guy exon 1. This is exon 2. And this is exon 3. Some genes have very few exons, 1 or 2. Some have 8 or 9 or 10 or 11 exons. What's going to happen? The introns ultimately will be spliced out of the mRNA once it's made. Right? Here it's DNA. Nothing happens to the introns in the DNA, but introns will be spliced out of the mRNA. Okay? What else haven't we decorated? We still have our 5' untranslated region. Okay? That's part of the DNA that gets made into mRNA, but it doesn't code for proteins, and it is not removed from the final transcript. And after the last intron here, we have the 3' untranslated region. And somewhere inside of that, we have our termination sequence. Okay, so now I've told you about almost everything in eukaryotic genes that you need to worry about. But there's one more thing that you really need to worry about. And that's here. The enhancer. So because we don't use sigma factors to regulate where RNA polymerase binds to DNA, we have to use something else. And we use enhancers. All eukaryotes do. Okay. Well, what's going on in our promoter? We'll come back to enhancers, but what's going on in our promoter? Our promoter is not all that weird. Separated into a couple parts. One of them is called the Tata box. And the Tata box is in about 25% of human protein coding genes. So I'm just going to put human genes. Okay. Just upstream of the Tata box is something called the BRE. This is called the B response element. So right now you ought to know what happens at promoters in general, and I hope you do. that RNA polymerase is going to bind a promoter, and it's going to bind this promoter knowing that there's a Tata box sequence and that there's the B response element. Okay? But this is a very complex set of things happening, much, much more complex than in prokaryotes. Why? Well, the B response element is really important, and it recruits these proteins called transcription factors. And the B response element primarily recruits 2 different transcription factors accompanied with about 10 other proteins that are all bound to them. And these transcription factors are what we call TF2B, which is from the B response element, as well as transcription factor 2D. Okay? What's cool about that? Well, what's cool about it is that either one of these guys can bind RNA pol 2. So these transcription factors will help recruit RNA polymerase to this area. Okay? The sad thing for them is that they're not very good at it. They can pull RNA polymerase in, but they don't bind very tightly. They don't do a good enough job to turn on transcription by themselves. And that's probably good for us because we have 20,000 genes in our genome and we need a higher level of regulation than just binding to the B response element. Okay? So what else do we have? Well, we have a Tata box. The Tata box has a consensus sequence. Let me write that on top. TA, TA, A, A. Heck yeah, that's right. Sorry. You're going to have to cut that out too. So this is the consensus. For the Tata box. But the Tata box sometimes is not there. It's only in about 25% of our genes. And so what's really important is that you know that none of these rules about transcription are 100%. But it's a really good model in that we understand how the Tata box works, and we understand a lot about how to recruit RNA polymerase to that. Okay? So what happens here? At the Tata box, what we really do is bind this protein called the TBP, the Tata binding protein. And this guy is bound to a lot. So sometimes you'll see this called TBC, the Tata binding complex. So this is a complex. Okay? And so one way to make sure that transcription in eukaryotes is highly controlled is that it's what we call combinatorial, where binding transcription factors that somewhat recruit RNA pol to the B response element is important. Tata binding complex binds to the Tata box. Now, all these guys bind to a whole bunch more proteins. These also bind, and what I always call recruit RNA polymerase. Okay? So you need a feature like a B response element. You need a feature like the Tata box. And now, I'll draw the Tata binding complex in blue. And now we're about ready to start transcription. But even with these two things, it's not really enough. So although each of these can recruit RNA polymerase, and RNA polymerase can be prepared to be turned on at the +1 site and start transcribing, It's not all the way done. Okay? So what are we missing? I'm going to move over here to stay in the camera. What we're missing is the enhancer. Okay? So, what you'll notice about this, if the Tata box is in 25% of the genes, and B response element is in a whole bunch of genes, that these two things are not specific. These two things are general. So we call these transcription factors general transcription factors because they're on lots of genes. But we don't always want to generally do things in eukaryotes. In fact, oftentimes we don't. So what do we have? Well, upstream of the Tata box and the B response element, which are in a promoter, way upstream at like minus 100 bases or minus 1,000 bases or minus 10,000 bases or minus 1,000,000 bases or minus 5,000,000 bases or somewhere so far away that you can't even believe it is an enhancer. Okay? And enhancers are sequences of DNA that bind specific transcription factors. Not general transcription factors like TF2B or TF2D, but specific transcription factors. And these guys can either upregulate, so turn on transcription, or they can downregulate transcription. Okay? How does this work? Well, transcription factors that bind the enhancer. So if I pretend like this box is my enhancer box now. Here's our enhancer. Transcription factors that bind to the enhancer, transcription factor will be in blue, are what we call modular proteins. So they have a couple things, a DNA binding domain. Okay? So they bind to the enhancer. They usually have what you might call a linker domain. It just provides some space so that you can do your other major job, which is carried out by a protein interaction domain. And these interaction domains bind to RNA polymerase. Okay? So, to make all this stuff come together, we now have a B response element that's recruited general transcription factors that are somewhat good to recruit RNA polymerase. We have a Tata box, which somewhat recruits RNA polymerase. But now downstream, we have an enhancer that is bound to a transcription factor. So your enhancer, bound to a transcription factor that is bound to RNA polymerase. And you might be saying, and rightly so, Casey, that's a long way away from the gene, and it seems like you're right. But if you take this out of the board, if you take it out of a two-dimensional context where I'm using markers, and you imagine that you can put your +1 start site right here, exons down here, there's your gene, here's your promoter, that the DNA can bend and place the enhancer right next to the promoter So these guys are trying to recruit RNA polymerase, the B response element, the TATA binding proteins, and the enhancer with its attached specific transcription factor, bringing RNA polymerase here. All of a sudden, we have a hugely strong combinatorial signal that the B response element, the transcription factor 2B and 2D want RNA polymerase here, the TATA binding proteins want RNA polymerase here, and the specific transcription factors bound to the enhancer are bringing RNA polymerase here. All of this creates a super stable position so that the RNA polymerase can start transcribing. Okay? This is most of the story, but we're going to get some more of it coming up.